Test, 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 test. Test, test. In the beginning was the word. Hello. Hello. Well, we seem to be a little thin on the ground today. And I'm not sure why. We're missing 1, 2, 3, 4, 5, 6 people, 7. And on Zoom, it's just me, myself, and I. So I'm not sure where folks are. It's 7:45, so it's time to begin. So we'll begin. So the immune response, the immune system, is a lot like the military. When you consider the military, you have one branch that is fairly nonspecific. It strikes hard, it strikes fast, but it's not very precision. And it has no memory. That would be the innate immune response. The innate immune response. So it strikes fast. It's very nonspecific. And it has no memory. Then you have another branch of the military that tends to be more surgical, is far more planned, takes a much longer time to get going, and has memory. That would be your adaptive immune response. So this is slower, much more precise, and has memory. So when you get a vaccine, which one is the vaccine trying to develop? Is it trying to develop a fast response that has no memory? Or does it take a longer time? And is it endowed with immunological memory? Okay, I saw somebody point at something. Can you tell me what you were trying to point at? Adaptive, exactly. Measles, mumps, rubella. Does measles, mumps, rubella protect you against malaria? No. M does not stand for malaria. It's specific. It's precise for measles, mumps, rubella. How long does it take you to get a robust antibody response against measles, mumps, rubella after you've had your first shot? Do you get it right away? Okay, I see head shaking over there. That's correct. About how long? Give me a best guess how long it takes. Close? About 2 weeks. About 2 weeks. That's a pretty good guess. So if you have a patient comes in and they want, say, a flu vaccine, you give them the flu shot, and you're going to tell him or her, this will take about 2 weeks, ballpark. It's going to depend on the person, on their health, their nutritional status, their particular genetic predispositions, things like that. But 2 weeks is a very good ballpark. Take about 2 weeks to get a good, solid immune response against this flu antigen. And that will give you memory so that if you experience those flu strains, you'll now have a secondary response that is robust and is faster than the initial response. And it will keep you from getting, if not the flu, it'll keep you from getting severe flu and what have you. So today, we're going to talk about this part of the military response, the innate immune response. So when you hear this, who's the first group you should go in? Well, it's usually the Marines. From the halls of Montezuma, you know, all that. So when you think of innate, just start whistling from the halls of Montezuma, the shores of Tripoli, and that will remind you of the innate response, I hope. Guys, immunology is very complicated. There are journals on immunology. People study immunology their whole life. They can't say that they understand the immune system. You guys need a working knowledge of the immune system. Some of the things we're going to cover are just bleeding complicated. So I will do my best to try and give you kind of the Reader's Digest version. I'll also apply you with plenty of my dumb mnemonics that might help you remember. But there's nothing about this that's simple. All right? Nothing about this that's simple. So if you're struggling with this, don't get down on yourself. Everybody struggles with this. I struggled with this when the floodwaters receded and I first learned immunology many decades ago. And then the field is rapidly changing. Still nobody there. Oh, well. So the innate, as we said, not specific, no memory. Adaptive is specific. It's specific for these particular things called antigens. Let's develop that term, antigens. Boy, blue is not my friend. Antigens. Foreign substance that is recognized by the immune system so that the immune system mounts a response to it. A foreign substance recognized by the immune system such that the immune system mounts a response against it. So let's say this is an antigen. Now, let's pretend that these two colleagues of yours are the immune response. So now, if I show it to you, this, can you recognize it? The antigen. If I show it to you this way, could you recognize it? Now, what about if I show it to you? That's a little tougher, but you're still going to have to learn. That's what if I show it to you this way. Now, why? Because antigens can give the immune system different looks. Those different looks will show you different parts of the antigen. Those different parts of the antigen that the immune system will make specific response against, those are called epitopes. Epitopes. So antigens, big. Epitopes, small. So this is an antigen. Point to an epitope then on the antigen. Okay, the spine would be one. Clarissa, point to another one. Okay, the bottom. Haley, point to another epitope on my antigen here. The cover, can you point to another antigen? Epitope, sorry, on my antigen, okay. The front look, okay. Is there another epitope we haven't quite gotten to yet? The top, yeah. And is there another one? We haven't quite got, oh yeah, the other side here. So every antigen usually has multiple epitopes. Multiple epitopes. And the antigen is big, it's foreign. The immune system says you're foreign, I'm going to mount a response against you, a piece on it that your immune system might specifically respond to. That's called an epitope. Epitope. Now, antibodies are secreted glycoproteins that specifically bind to and antigen. They specifically bind to, recognize antigens. Now, when they bind to antigens, they do two things usually. More than two, but two general things. Number one, they will tend to neutralize them. They will tend to neutralize them. So if that antigen, say, is a toxin, by binding the antibody to the toxin, neutralize it. Two, when you bind an antibody to the antigen, you now mark it for destruction. It's very easy for the body to get rid of something that's antigen tagged. So essentially puts, I know none of you guys did this, but used to be a really mean trick to pin something on the back of somebody, a sign that said kick me, and everyone would be kicking them. They had no idea why. The sign was on their back. It said kick me. That's kind of what you do when you bind an antigen to an antibody to an antigen. Put a tag on it that says, get rid of me. Get rid of me. So that is an antibody. That's what antibodies do. Now, who makes antibodies? B cells. B cells. And they are part of the adaptive immune response. B cells also will show immunological memory. And that's why you can have some diseases and be immune against them for the rest of your life. Others, not so much. And we'll just kind of hint at why that might be the case. Very good. Any questions on any role of this before we move on? No? Okay, then. Here are some of the elements of the innate immune response. First of all, physical barriers. Physical barriers very simply get in the way. Skin would be the biggest example of a physical barrier. This is one of the reasons why burn patients are at such high risk for disease and for dehydration, because skin represents probably the most robust barrier against infection. Then, of course, mucous membranes that coat the inner surfaces, that coat your buccal cavity, that coat your nasal cavity, that coat your GI tract, so on and so forth. These are physical barriers as well. They just get in the way. These are designed to resist penetration. Then you have chemical barriers. Chemical barriers, very often used in combination with physical barriers. So chemical barriers would include things like salt. This is particularly used in your skin. Your eccrine sweat glands secrete respectable amounts of salt and ammonium. Then they also secrete lipids. And then bacteria on your skin cleave those lipids. They produce fatty acids. And those fatty acids drop the pH of the skin. That makes skin relatively inhospitable, except for the organisms that colonize it, mainly staph and other organisms like that. So salt, pH is also a big factor in your stomach as well. Lysozyme is everywhere. It's in tears, it's in sweat, it's in saliva, it's everywhere. Lysozyme degrades peptidoglycan. It degrades peptidoglycan, which is the major component of bacterial cell walls. And this enzyme literally is everywhere. Transferrin. What's transferrin? You might remember that from last year, your A&P class. Anybody remember? Look at that ending, ferrin. That remind you of something? Ferrin? Ferrous, ferric. iron. This is the major iron-carrying protein in your blood. Iron is an oxidizing agent. Do you want iron floating around free in your blood? Survey says no. No, that's a one-way ticket to major league cellular damage. So transferrin binds iron, carries it in the blood. Transferrin binds iron fairly tightly. If organisms therefore get inside your body, they will find themselves in an iron-limited environment. They can't grow without iron. Transferrin essentially prevents them from using excess iron, from being able to scavenge iron. Transferrin says, nope, my iron, my iron. Lactoferrin. Lacto, that sounds like guys, with your mask on, it's hard to hear. I need you to speak up. Lactose, which is a major sugar in milk. So this is an iron-binding protein in milk, in breast milk. Once again, if you have organisms that colonize the areola of the breast, and they're trying to live off fats, proteins, sugars, and milk, they won't get much iron, because this is tying up the iron. That will limit growth in milk. That will limit growth in the lactiferous tissues of the breast. And then defensins. Defensins are in blood. They're in mucus. They're in tears. And they bind to the surfaces of bacteria, and they put holes in them. So these are antibacterial peptides, small peptides. Now, there is another one that's not on here that I think is worth an honorable mention, and that is mucus. And we'll do mucus in green. Mucus is made by goblet cells in the nasal, respiratory, and GI tract. In the duodenum, they're made by Brunner's glands as well. Brunner's glands are deeper. The goblet cells are in the epithelium, Brunner's glands are beneath in the submucosa. Mucus coats the apical surfaces of cells. Mucus is made of very highly charged sugars. So mucus is very, very wet. Takes up long. Mucus also creates a very, very difficult to penetrate barrier. Mucus is also laced with lysozyme. It's laced with transferrin. It's laced with antibacterials. It's essentially a briar patch full of landmines for bacteria. And if bacteria want to invade that mucosa, it has to deal with that briar patch first. So I went hunting once. When I was younger, I used to duck hunt. I was never very good at it, but I enjoyed it. And we went duck hunting one weekend, and my friend, he sent several of our decoys to the bottom of the water. Oh boy, those decoys are expensive, and he had a thing for those decoys. Anyway, we did get a duck, but it veered and then off to the side. We said, okay, we had a dog. Let's go send the dog to get it. And he just sits down there. And he's sitting there, and we're saying, well, go get the dog. The dog kind of looked up at us as if to say, are you kidding? And it was a briar patch. And we really wanted that duck. You ever stumbled into a briar patch? Excuse me. thorns that pull at your clothes and put holes in them. They prick your skin. Many of them are full of little sacks of irritating chemicals that irritate your skin, cause you to itch and burn. And then the leaves, some of them, if they get on your skin, they cause you to itch and burn and stuff like that. We spent about 5 minutes in there, and then we said, we're defeated. So the dog knew better than we did. And that's essentially, that's what bacteria have to negotiate if they're going to invade a mucosal surface, is mucus. Now you have other defenses. We'll talk about each of these. You have cells also that are designed to clean up unwanted debris, of which bacteria is one of them. You have an inflammatory response, a fever response, and then you have particular molecular defenses that we'll talk about as well. So physical, chemical defenses, then you have cellular defenses. Now this slide just lists the different cells. I would prefer to show you them. So I'll show you these, then we'll come back and talk a bit about dendritic cells, because those are important. So this slide actually gives you a picture of what they look like. I think if you can equate in your mind what they look like, you might remember what they are a little better, rather than just seeing words. So this stain, this is called a hematoxylin and eosin stain, or as it's mercifully abbreviated, H&E. Now, of the E there, eosin changes colors according to the pH. And the white cells are classified according to, number one, whether or not they have granules or whether or not they don't have granules. So these cells here have granules, these large granules here. These don't have detectable granules. So these two types of cells are called agranulocytes. Agranulocytes. These have granules, so they are granulocytes. Let's take a look at this one here, the neutrophil. Now, eosin, if it is put in a basic solution, an alkaline solution, eosin turns purple. And if you look at these granules, what color are they? Purple. So these have basic granules, so they're called basophils. Basophils. The granules are basic, so they like basic pHs, so they're basophils. Basophils. Oops, I'm over here. Sorry. Basophils. Wrong picture. Basophils. Lots of purple granules there. These here, eosinophils, when eosin gets in an acidic environment, it stains cherry red / pink. What color are these granules here? Pink. Pink. So these have acidic granules. They like the eosin, so they're eosinophils. They like the eosin, eosinophils. Then these cells have granules that are kind of gray, gunmetal, boring. They're neutral pH, so they're called neutrophils. So basophils, eosinophils, neutrophils, those are your three types of granulocytes. They have granules, 3 different staining patterns when you stain them. Eosinophils play a role in allergies, and they play a role in parasite infections. If someone is infected with a parasite, their eosin levels will climb pretty precipitously. When you look at the worm or something like that in their body, they're surrounded by eosinophils that are trying to kill it. Also, some people with severe asthma will have lots of eosinophils in their blood. So, patient comes in for a physical, you send them to the lab, draw blood, or you draw the blood yourself, send it to the lab, comes back, their eosinophils are high, means one of 2 things. Means they have allergies, bad allergies, or they may have a parasite infection. And in the United States, at least, allergies are probably the safer bet. But what if they're an immigrant and they came from a country where infections like that are endemic? Then you might not want to make that assumption. Neutrophils. Neutrophils are the sprinters of the innate immune response. They're the first to show up on the scene. So like the Marines, they're the first to hit the beach. They are very, very active white cells. These cells are called professional phagocytes. Professional phagocytes. They gobble up cell debris. They gobble up dead cells. They gobble up any invading bacteria. And they do it vigorously. And in fact, they do it so vigorously, they'll burn themselves out very quickly. So these cells take the candle and they light it at both ends, and it burns, and they essentially, they put it all on the field, shall we say. Basophils. Basophils tend to be found in tissue. They don't tend to, once they get to their spot in tissue, they don't tend to move very much. The granules in these cells are filled with compounds that will play a role in inflammation. We'll talk more about those compounds, but to dispel the suspense just a bit, they contain something called histamine. We'll talk about the role of histamine here in a bit. And then other things called leukotrienes. And some other stuff too, but those are the main things in their vessels, in their granules. They will play a role in inflammation. We'll get to that in just a bit. The agranulocytes, monocytes. Monocytes are the biggest of the white blood cells. Monocytes, when they are immature, will tend to have a nucleus that's either roundish or might have a little indentation in it. Monocytes, then, when they're activated, they have a nucleus now that gets very lobulated, and they become known as macrophages. Macrophages are also a type of professional phagocyte. However, if the neutrophil is the sprinter, the macrophage is the distance runner. The macrophage will not burn itself out nearly as fast as a neutrophil. Also, the macrophage plays an important regulatory role in the immune response. So it's a regulator and a professional phagocyte. So it's like the lieutenant in the platoon that's also one of the best sharpshooters. Something like that. Lymphocyte. Lymphocytes come in three types. Three types. There's B bodies. There are also, where did I put my black marker? There's T lymphocytes as well. These play a huge role in regulation and other things. And there are natural killer cells. And those are the third type of lymphocyte. Those play a very important role in cells, dead cells, dying cells, and cancer cells as well. Now, dendritic cells. Here is a picture of some dendritic cells. Dendritic cells have all these extensions of the membrane. When they're born in the bone marrow, they're actually born as monocytes. They then migrate throughout the body. Then they take up residence, usually in some part of the body, the nasal mucosa, GI tract, lungs, wherever. These guys are sentries. Sentries. What do? They sit there and they survey. They're constantly taking up material and they're ascertaining if that material is foreign. If that material is foreign, they will present it. So they do something called antigen presentation. Antigen presentation. Think of it this way. Let's say you had to let's say you had to go through and get rid of all the kids' toys that were exposed to, say they were very sick, and you had to go through and get rid of all the toys that probably got exposed to the germs that made them sick, and you had to just really take them out and burn them. How would you know which ones needed to be burned? You need to have them boxed and presented to you as toys that you were to burn. That's what antigen presentation does. It essentially licenses the immune system to identify something as foreign and mount an immune response against it. Dendritic cells are the major antigen presenting cells in the body. They're not the only cells that present antigen. They're definitely the major ones. From whence do these cells cometh? Well, they come from the bone marrow. Now, in the bone marrow, mainly the bone marrow of the pelvis, ribs, and sternum, you have a stem cell population. This slide says it's a pluripotent stem cell. That is incorrect. That's wrong. I wish books would stop doing this. Pluripotent means it can become any cell type in the adult body. That's wrong. That's incorrect. These cells cannot become any cell in the adult body. They can become a subset of cells in the adult body. Therefore, these cells are multipotent. They're not pluripotent. And I wrote to Professor Black and told her, please don't misuse this terminology. We have enough confusion in the literature without textbooks adding to it. And she said, well, that's what some papers say. And I said, the papers are wrong. And there have been debates in the literature. And here are the papers that discuss it, why this is wrong, blah, blah, blah. And I haven't heard back. But anyway, suffice it to say, they are multipotent stem cells. If you call them pluripotent, you will get it wrong. So just scratch it out in your book and write multipotent. And no, I'm not going to let this go. So this multipotent stem cell is called a hematopoietic stem cell. Good night, that's a mouthful. So they divide and they will make a daughter cell. Divides, one cell stays a stem cell, the other daughter cell now becomes either a lymphoid progenitor well, no, that's the daughter cell becomes or the daughter cell can become a member of the myeloid lineage. Myeloid lineage. Now, the book here says that it becomes either a lymphoid progenitor or a myeloid progenitor. There were 2 papers in Science, the journal Science, 3 years ago that called into question the existence of this cell. So the existence of this cell has not been rigorously shown, and there's evidence to suggest that this cell never forms. In fact, what probably happens is that once this daughter cell divides, it's already committed to becoming a megakaryocyte or granulocyte lineage or straight to the erythroid lineage. So it might go daughter cell either to this or to this or to this or something like that. So this cell may not exist. Just something to keep in mind. But the 2 lineages do exist. The myeloid lineage consists of red cells, the megakaryocytes that give rise to platelets, the granulocytes, and then also the monocyte lineage as well. Then there are the lymphoid cells. The lymphoid lineage, the lymphoid progenitor has been demonstrated rigorously. That's the best. That cell then will give rise either to a B cell, which will mature in bone marrow and then be released into the peripheral circulation. The T cell that has to mature in the thymus, a gland over the heart, or a natural killer cell that also matures partially in the bone marrow, partially in the peripheral circulation. How do these get turned to this, to this, to this, to this, to this, whatever? The answer is a collection of signaling molecules. These signaling molecules are called cytokines. Cytokines. So let's say here, Victoria wanted to pass a message somehow to Kagan down there. She didn't want to get up and walk over and tell her in the middle of lecture. So let's say Victoria just pulls out a 3x5 card and just writes on it, oh, the cookies last night at dinner were really good, weren't they? Or something like that. Then you pass the note to Anna, and then Anna passes the note here, and then you guys read the note, and you look over at Victoria, and you go, no, yeah, those cookies are pretty good. And you pass it to Karis, and Karis passes it to Kagan. Kagan then gets a note, and all of a sudden she really wants a cookie. Now, what has happened? You have sent a signal to Kagan from a distance. That note would be like a cytokine. That's how cells pass notes to each other. They use cytokines. Cytokines are small peptides. They diffuse very quickly. They don't tend to have long half-lives. They can, in some cases, signal in an endocrine fashion. That is, they can be released into the bloodstream and then signal to tissues that are far away from them. But most of the time, they signal short distances. That's called paracrine signaling. Paracrine. Now, other times, cytokines will signal to the cell itself that is secreting it. So let's say, go back to our example with notes and Victoria. Let's say Victoria had a haircut today, but she's a very busy lady. She's a nursing student. That just means she's busy all the time. She might forget. So what she do? She writes in a little note and she pastes it right there. So when she studies, she opens that. She says, oh, I have a hair appointment today at 2:30 or something like that. Then she opens that up and says, 2:30. She wrote a note to herself. So sometimes cytokines benefit or signal to the cell that's actually making them. That's called autocrine signaling. Autocrine signaling. A-U-T-O-C-R-I-N-E. Autocrine signaling. So we have paracrine, short distances like from Victoria to Keegan. We have autocrine, that's like the note, you have a haircut today at 2:30, or endocrine. Endocrine. So Anna writes a note and then she dumps it in campus mail. And it goes all the way to the house. And it tells Professor Ulrich, congratulations on the engagement of your son or something like that. And your daughter, so on and so forth. So that would be endocrine. Signal long distance because it's dumped in the bloodstream. Cytokines can signal in all three ways. These cytokines tend to signal short distance. Now, the cytokine proportions and cytokine concentrations will influence what type of cells these will become. Let's just talk a little bit about a few of these cytokines. So there is a cytokine that's called granulocyte, monocyte, colony, stimulating factor. I don't want to abbreviate that. GMCSF. That is a cytokine that you find that will shunt daughter cells into this lineage here. So you make more basophils, more eosinophils, more neutrophils, more dendritic cells, more monocytes. Then there's another one that's called granulocyte colony stimulating factor, or GCSF. G-CSF stimulates the production of neutrophils. Then you have another one called erythropoietin. Erythropoietin. That's made by the kidneys. It's made by the kidneys when your red. This will shunt cells into the erythrocyte lineage. And then you have thrombopoietin. Thrombopoietin. Thrombopoietin will push cells to become platelets. Platelets bud from this larger cell called the megakaryocyte. They just blood from the cytoplasm. And thrombopoietin will push cells straight into the platelet lineage. These are examples of cytokines that will direct. There's many more. This is just a few. Some are very general. Interleukin 1, interleukin 3, general growth factor, stem cell growth factor, that sort of thing. Others are more specific, like these, that push cells into particular lineages. That's how these are born. Again, they're born in bone marrow, but they may in some cases need to go elsewhere to finish their maturation, as in the case of T cells. They have to go to the thymus. Very good. It's break time. Let's go ahead and take a break. Take 5. Refresh yourself, and then we will continue. Mm. Yeah.
So about the emailing, I signed up for health school at Jackson Christian, and I get done at 7:45. Would you rather me come late on Wednesdays, or would you rather?
Well, probably Wednesdays run in on Zoom,
Oh, okay.
so you can just Zoom in.
Oh, okay.
In fact, if you're at Jackson Christian, although I've heard the wireless at Jackson Christian is spotty in places, but there are places apparently where it's pretty good.
Okay.
Mrs. Schaefer's husband, Jim, works there. He'll know all the good spots.
Okay.
You could probably just stay there. That's a bit of a drive
That's like,
to go from there to here.
it's like 15, 20 minutes.
Yeah, you could probably just stay there
Okay.
and do it. There are plenty of places you could probably just sit there in a quiet space and
Okay.
not be bothered, I would guess.
Okay.
Because their enrollment is down. Yeah, yeah. So okay,
Yeah, I can.
that'll work.
Okay.
That'll work. Okay, my clock says it's time to start. Let's talk a bit about intracellular killing by professional phagocytes. How does a phagocyte know the difference between an invader and a part of you? In other words, how does it distinguish between friend and foe? Between friend and foe. Pathogens possess what are called pathogen-associated molecular patterns, which are mercifully abbreviated PAMPs. PAMPs. So if we brainstorm, here's what consists of PAMPs. Peptidoglycan. Do our bodies contain peptidoglycan? No. That's a molecular pattern. Therefore, if our cells see it, they know it's foreign. It didn't come from us. Make sense? Lipopolysaccharide. Is that found in our cells? No. So if it sees it, it being a phagocyte, if it sees it, then it will know that it is foreign. Flagella, bacterial flagella proteins. Do we make those? No. So if your cells see it, they know it must have come from a bacterium. So on it. So these are the types of molecular patterns that cells essentially will detect that are pathogen associated molecular patterns, or PAMPs. There are, on the surface of phagocytes, receptors for PAMPs that are called PAMP recognition receptors, or PRRs. PRRs. I apologize for the terminology. I don't make the news, I just report it. So don't shoot the messenger. It's just the terminology, I realize, is formidable. Just, the abbreviations are fine. Just know what the abbreviations mean. Know what? So PRRs are receptors for pathogen-associated molecular patterns, PAMPs. PERS and PAMPs. The PERS bind the PAMPs. And my immunology professor, Eli Benjamini, he was from Israel. He had a very thick accent and he spoke really fast. He used to talk about, think about the cat sitting on your lap and you pet the cat and it purrs because it's captured your hand. Your hand is the PAMP petting the cat and the cat now purrs. PAMPs, purrs, okay. That was his way of helping people remember. Class like this, student piped up and said, yeah, but I hate cats. I said, well, cats are pathogens there. That helps you. Because you hate them, that's part of the whole thing. Anyway, so the PRRs, the PERS, come in two main types. There are those that white cells use to clamp onto the cell and gobble it down. They're used for phagocytosis. There are others that are used to produce a signal in order to, excuse me. There are others that produce a signal that revs up the immune response. Now, there's a logic to this. Think through this with me, if you will. Let's say you live on a farm and you have chickens. Chickens occasionally will get on the roof of your house and will stomp away on the roof of the house, and you're convinced someone's up there trying to break in. So if you call the police are going to come out and they're going to propose that they arrest your chicken, and that would be a bad idea. Now, what should you have done before you called the cops? Should have gone outside and checked, right? That's what these phagocytic receptors allow the phagocyte, because when you activate the immune response, you're going to activate some tissue damage. If you activate tissue damage, that's going to now trouble the host more than it should. The phagocytic receptors let you clean this up without making too big a fuss, essentially. So the phagocytic receptors come in two types: opsonizing, non-opsonizing. So let's talk a bit about an opsonin. Opsonin. This was illustrated to me once when a friend of mine who was Arab invited me over to his house. And in Arab houses, authentic houses, you take this hand and you sit on it, you eat with this hand. Now, they serve meat that has a lot of sauce on it, and things get pretty slippery. So sometimes they give you these little balls of rice. You try to pick them up, and it goes. You chase the thing around your plate, and pretty soon you have to just kind of smash it and then pick it up and eat it like that. That's kind of what it's like for a phagocyte to chase around a bacterium that's got a capsule or something like that. It's slippery. It's like trying to pick up a Italian meatball with slippery hands. You can't pick it up. What do you need to pick it up? A fork. You spear it, and now you can eat it without much ado. That's what an opsonin is like. It's like a fork that helps you pick up the greasy Italian meatball or a Swedish meatball or, whatever. One of the best opsonins that we will see are antibodies. Antibodies are excellent opsonins. And there are groups of receptors that are opsonizing, phagocytic receptors that are receptors for antibodies. Others are non-opsonizing. They don't require something to opsonize the cell. There's a whole group of these, and what they look for are things specific to bacterial cells. So, for example, the sugar mannose. Mannose is relatively rare in our own glycoproteins. We don't have a lot of mannose. Fungi and bacteria have a fair amount of mannose. So some of these receptors bind mannose. And if they bind mannose, they will use that binding to take it into the cell. Secondly, there are another group of receptors that bind a lipid that is found in bacterial membranes called phosphatidylserine. So these you can just remember as PlayStation receptors, PS. So think, kill the Space Invaders, whatever. And there's some others. There's a group of receptors called scavenger receptors that find little molecules on the surfaces of various bacteria, acetylated lipids, things like that, that you find in bacteria, you don't tend to find in our bodies, and it will bind them and use them to gobble them up. Now, once they gobble them up, you get a deformation of the membrane. The membrane then extends around the body. The membrane then will fuse. You then make a vesicle inside the cell called a phagosome. Phagosome. Once you form a phagosome inside the cell, the phagosome will fuse with a lysosome. That produces a phagolysosome. The lysosome contains lots and lots of nasty things that the cell will have a rough time survive. The bacterial cell will have a rough time surviving. I think on the next slide, we'll be able to go over those. Yes, we will. We'll go over those when we go to the next slide. Let's talk a bit about the types of cells. So neutrophils, monocytes, macrophages. Again, neutrophils show up first. Macrophages then later. And monocytes, if they show up, they become activated. then they become macrophages. Now, macrophages come in two types. This is important. There is a so-called M1 macrophage and an M2 macrophage. The M1 macrophage is very much a phagocytic macrophage. It will phagocytize and kill bacteria. And it signals for other cells to come, and it's a pro-inflammation cell. M2 macrophages are more associated with tissue healing. And the macrophage comes from the monocyte. Again, they appear second after the neutrophils. Very good. Let's talk a bit about the killing that goes on inside the white blood cell. So the phagolysosome has two types of granules. Two types of granules. There's a so-called specific granule. A specific granule and an azerophilic granule. Azerophilic. So the specific granules tend to go to work first. They contain enzymes that degrade macromolecular assemblages. So they contain proteases and hydrolases. And why do these act first? These act first because when the phagosome first fuses with the lysosome, the pH inside is neutral. So these are active at a neutral pH. And these contain proteases and hydrolases. Proteases degrade proteins, hydrolases degrade just about everything else. Lipids, polysaccharides, things like that. These are active at a neutral pH. As the phagolysosome matures, it becomes more acidic. As it becomes more acidic, the azerophilic granule will now take over. So this is more active at an acidic pH. And this contains very powerful enzymes, one called cathepsin. Cathepsin G, and then it contains a group, well, it's a complex that will make something called reactive oxygen species. It's called myeloperoxidase. Myeloperoxidase. Now the enzymes will kill about 2% of the pathogens. The reactive oxygen species made by myeloperoxidase kill about 98% of the bacteria that the thing gobbles up. So you get a respiratory burst you have an NADPH oxidase. This will take molecular oxygen and it will convert it to things like superoxide radicals, to things like hydrogen peroxide through chemistry called the Fenton reaction. These react with iron to produce hydroxyl radicals, hydroxide radicals that are very, very damaging to cells. You also have chloride that's produced. Chloride will react with these to make hypochlorous acid. And then if that wasn't bad enough, you have an inducible enzyme called nitric oxide synthase. Nitric oxide synthase takes the amino acid arginine and it converts it to citrulline plus nitric oxide. Nitric oxide will react with these reactive oxygen species to make something called peroxynitrile radicals. Those are devastatingly deadly to cells. So basically, you have a little compartment in the cell where you make every nasty molecule known to cells and just concentrates them inside there. And the bacterium basically goes, it's essentially like putting the bacterium down and just beating on it with two fists tied to bricks. It's not pretty. So this is very effective at killing bacteria. Very effective. So you make superoxide, you make hydrogen peroxide, nitric oxide, chloride will give you hypochlorous acid, and then the nitric oxide reacts with these to produce peroxynitrile radicals. Peroxynitrile. Very, very nasty. Very nasty. So that is the result of the oxidative burst. This oxidative burst is so good at killing things that it eventually kills the white cell with it. That's why your neutrophils are really good at this. That's why neutrophils burn their candle at both ends. That's why they die so quickly. They essentially give their lives for your health. A moment of silence. Okay, that's enough. And I think you get the point. Now, here's the other thing. Can those potentially cause a lot of tissue damage of the good tissues? Yeah, they can. That's why inflammation can cause local tissue damage. That's why chronic inflammation can be such a terrible thing for your body. Sometimes cells don't dissolve everything. They will have what's called a residual body that they then will get rid of, and they'll spit that out essentially. And these will be either degraded further by stationary cells, Kupffer cells, on the liver, reticular cells in the spleen, or sometimes they're just packaged with bile and then secreted out to the GI tract or whatever. Are there questions about white cell killing, about oxidative burst, myeloperoxidase? Oh, do you guys remember from Pathophys, we talked about chronic granulomatous disease? Did that ring a bell? Chronic granulomatous disease is due to defects in the genes that encode myeloperoxidase. So echoes from another class that hopefully maybe make sense out of that. Very good. Now, check my microphone. Hello, hello, hello. Hello. Yeah, it's hearing it. Okay. Well, I'm gonna keep going then. Now, the lymph system does what? Let's think about this, try to review a little bit from A&P last year. So when the blood goes to the capillaries, on the arterial side, you have high pressure. The capillaries are just one cell thick, but the endothelial cells are not perfectly interfaced with each other. There's little tiny holes, so liquid will start to squeeze out of the capillaries. Liquid and a little bit of protein, not much though, no cells, small of a hole. So now the protein concentration in the serum goes up. You then circulate through the capillary, and that fluid, that becomes your interstitial fluid. That bathes the cells of the tissues. Now you get to the venous side, your pressure is low because you're not on the arterial side anymore. But the oncotic pressure in the vessel is high. So what does it do now? It draws fluid back in. Now, does it draw all the fluid back in? No, draws about 93% of it back in. There's still 7% of that fluid that's left there. You have to return that back to circulation. If you don't, your tissue is going to go get lots and lots of fluid in it. The lymph system does just that. It returns that fluid back to the circulatory system because the lymph vessels will now then merge into the thoracic duct. That will then merge with the subclavian veins, and it dumps that fluid back into the circulation. That's essentially what the lymph does. Lymph vessels are largely controlled by contraction of skeletal muscles and also by slight peristaltic contractions. And this is nicely illustrated. I don't know if any of you, when you get on the plane, if you take your shoes off, and then when you land, you put your shoes on, and your shoes feel a little tighter than they used to. That ever happened to any of you? Yeah, why not? Because you're not walking, you're not pumping the lymphatics. So fluid will tend to build up in your foot swells a bit. Therefore, when you put your shoes on, now they're a little tighter than you remember them. Once you get off the plane to the luggage carousel, that goes away. You're pumping the lymphatics. The lymph vessels also have little way stations called lymph nodes. Those lymph nodes are loaded with B cells, T cells, and dendritic cells. They are surveillance way stations. They are part of the watchtowers along the wall, if you will. They are going to determine if any foreign stuff has found its way into the lymph. And then you have primary lymphoid tissues, thymus, and the big one is a spleen. Then you have other secondary lymphoid tissue, not just the nose, but also the gastric-associated lymphoid tissue, tonsils, you know, whatever. There's lots of different types of associated lymphoid tissues, both in the nasal mucosa. There's a mucosa-associated lymphoid tissue, MALT, gastric-associated lymphoid tissue, GALT. These are concentrations of lymphoid tissue where you have mucous membranes, because those are areas most subject to assault by organisms. Now, if you look essentially at the lymph node, you have vessels that come in, you have valves, that's to make sure the flow into the lymph node is one way. You don't have a heart that pumps through the lymph, it's all very indirect. The lymph node has a cortex on the outside, the outer portion of it at least, and then a medulla on the inside. The cortex contains, on the outer part at least, will contain macrophages, B cells, and follicular dendritic cells. In the inner cortex, you'll start to have T cells and dendritic cells. And then in the medulla, you have B cells, plasma cells, and macrophages. Essentially, antigens will first experience dendritic cells. The dendritic cells will present the antigen to macrophages, B cells. As the antigen makes its way further into the lymph node, now it encounters macrophages and, T cells. These dendritic cells now will present the antigen to T cells. Plasma cells are mature B cells. These mature B cells secrete antibodies. They secrete antibodies. They secrete antibodies because they've had an antigen presented to them that they recognize, and now they're making antibodies against it. As you get then into the interior, into the medulla, you then have these cells that fully recognize the antigen. Those cells will then leave the lymph node, and then we'll be able to take it to the peripheral circulation. All right, inflammation. There are 4 classic signs of inflammation: redness, heat, swelling, and pain. Redness, heat, swelling, and pain. And if you use the Latin terms all rhyme. Rubor, calor, dolor, tumor. That's a nice way to remember them if you remember the Latin terms. Let's say Karis over there decides for Christmas she's gonna work out and show her family what a great athlete she is. So she goes to the gym and she hits the weights and she lifts and she lifts and she lifts and she's so proud of herself. Well, she wakes up the next day, she discovers she's paying for it. She's going to feel, come on, you've had this happen to you, sore, right? Why is she sore? Because she overused the muscle got damaged, there's low-level inflammation in the muscle. That's why she's sore. What caused that inflammatory response? That's what we'll talk about on Monday. And I hope you guys have a good weekend. We have a Bible quiz this weekend. Great for my quizzers. John 4, 5, and 6. That's a lot of scripture. So that's tomorrow. See you guys on Monday.